Thanks. Okay, well, thanks for opportunity to talk. I'll be presenting today on a sort of side project that I've been working on off and on for the last bit on demographic collapse of maize in the Americas. And maybe the subtitle should be several lessons in doing science, or how not to do science, something like that. And so the results are somewhat preliminary, and although I think that qualitatively we've got the right answer, as you'll see at the end, there's still more work to do. So we're all probably familiar with the impacts of European contact on the Americas, the resulting loss of life due to disease, due to enslavement, due to loss of land, due to war. So pretty dramatic impacts of European contact in the late 15th and early 16th century on indigenous populations in the Americas. And there's extensive historical documentation of this. And indeed, estimates, the overall estimates of what this looks like in terms of population size are something like this from a nice paper a couple of years ago. The estimate of the population of the Americas before the year 1500 is something around 60 million people across North and South America. So a fairly substantial population. And after post-contact, the population size drops to about 6 million. So that's consistent with about a 90% reduction in population size. And this, of course, varied in different areas. It wasn't 90% across all of the Americas, and wasn't important to keep in mind throughout this that what's been referred to as the Great Dying, or this loss of life, was not a single event that happened in one year, but the decline in population size took place at different times in different populations and to different extent in different populations. Nonetheless, overall, a 90% reduction in population size is pretty egregious. And population genetic data are also entirely consistent with this. So a number of authors now have looked at genetic samples from indigenous populations in the Americas and shown that across the board, there's a bottleneck. Let's see, can you see my, you can't see my cursor, here we go. Across the board, there's evidence consistent with a bottleneck right around the time of European contact. So on these graphs, this is generations in the past and effective population size. And so right around something like 18 to 20 generations ago, given a generation time of about 25 years for humans, that's consistent with a dramatic bottleneck due to European contact, or right around the timing of European contact. And that's been shown, as I said, in lots of different populations, although the extent varies among populations. And so a couple of years ago, reading some of these papers, I had the idea, well, if 50 to 90% of farmers perish, what happens to their crops? Was there evidence of a great dying of American crops? And here really is the first lesson of doing science here, is to never pay attention to other disciplines, because if you do, you will be disheartened to discover that they've figured everything out a long time before you did. And indeed, after a bit playing with the genetic data, I started looking at the archaeological data, and it turns out that there's widespread acceptance in the archaeological literature that this indeed has occurred, and there's considerable evidence that this has occurred. So this is just one line of evidence. This is from lake cores I believe in Panama, and shown here are years before present on the x-axis, and charcoal abundance in the lake core. And charcoal abundance is consistent with slash and burn agriculture. So indigenous peoples would cut the forest, burn it. That provides plenty of fertilizer and rich soils for crops, plant in that for a few years, and then move on to a new patch of forest and rotate this throughout forested regions. And so there's considerable evidence of charcoal associated here. This big spike is associated sort of with this one here is associated with the beginning of agriculture. And there's a precipitous decline to zero or near zero of charcoal in these lake cores associated with European contact. And concomitant with that, if you look at evidence of phytoliths or pollen, phytoliths are these small silica granules that are in microfossils that are found in plants, and they can identify the level of genus or species based on phytolith morphology. Both phytoliths and pollen evidence in these lake cores show the sort of opposite pattern, that after European contact, there's a huge rebound in both phytolith and pollen evidence of forest abundance, consistent with forest regrowth after arable land has been abandoned. And indeed, in several regions, if you look specifically, for example, for maize pollen, you can find evidence of a decline in maize pollen associated with European contact. So there's really widespread acceptance that, and you know, if you want the take home, this has definitely happened. The archaeological literature has shown that this has happened, and it was almost undoubtedly the case for many different American crops. Indeed, to the extent that a hypothesis put out a couple of years ago by Koch et al. Suggested that the extent of decrease of slash and burn agriculture and regrowth of forest was sufficient across the Americas, that enough arable land was abandoned and enough forest regrew to explain the observed decrease in carbon dioxide levels worldwide. So around 1550, 1600, there's this dramatic decline in CO2 levels worldwide. And this decline in CO2 actually led to a 0.1 to 0.2 degree Celsius cooling, global cooling around this time. And Koch et al. Have argued that that's due to forest regrowth in the Americas because of the great dying leading to such a dramatic abandonment of arable land, which regrew into forest. This hypothesis, the association between the forest regrowth and global cooling, is controversial. So Bush et al. Argue that some of the forest regrowth happened before European contact. But nonetheless, there's widespread agreement in the archaeological literature that there was widespread abandonment of arable land, and pretty obviously then a decline in farming. But as geneticists, we have basically ignored this. So if you look at both theoretical literature on domesticated crops and demography of domesticated crops, or estimated models of crop demography for Mesoamerican species, whether it's beans or even stuff that my lab has done in maize, we've sort of just ignored anything after domestication, and we assume that there is a bottleneck associated with plant domestication, and then everything's hunky-dory and the crop takes off and is spread everywhere and you have exponential growth, and ignored any sort of demography after the fact. So really, I was unable to find any evidence of, or any case of a Mesoamerican crop where someone had looked at, using genetic data, looked at demography sort of more recent than domestication. And so I set out the sort of goal of this, and what I set out to do is say, well, if we look at the genetic data, can we actually show in the genetics for maize or other species that there was a dramatic bottleneck associated with European contact? And the first way I went about doing this was basically just copying what some of the human geneticists have done. So Browning et al., for example, used patterns of identity by descent to reconstruct the recent past. And the basic idea is that you identify in genomes segments of identity by descent, or IBD. So this orange segment in here between these two genomes. And those segments are essentially bits of a chromosome, as you can show here, that were inherited from a common ancestor. So these two individuals in the present inherited that segment of the orange chromosome from a single allele in some common ancestor in the past. And the rate of that coalescence, or the rate of identifying IBD segments, is essentially the inverse of the population size. You'll find a common ancestor more quickly in a smaller population size, and it will take longer to find a common ancestor in a larger population size. So the number of IBD segments, the length of these IBD segments in terms of the genetic map, so how long they are in centimorgans, that pattern of chunks of identity by descent in a set of genomes will tell you things about the size of populations backwards in time. And so using this, I decided to take a look at some whole genome sequencing data from some Mesoamerican crops. And I had good whole genome sequence data from both maize and beans, and a genetic map from both maize and beans. Here I used 55 individuals from a single farmer's field because I was trying to worry about population structure. We phased and imputed these guys for the beans because they're homozygotes. You have to merge them into pseudodiploids. and then detected IBD chunks and merged them and estimated population size. And so using my best estimates of a number of these parameters, I came out with a model that looked really nice and supported and supports this idea of a bottleneck associated European contact. So at least in this model, maize population size is something like 500,000 in the past and declines to 60,000 after European contact. Bean population size goes from about 200,000 or maybe 150,000 down to 5,000 or 6,000. So there's a more than 90% decline in population size for both of these models, or for both of these species. I was hoping to be able to do this for a number of other species, but it also turns out that finding good data is really hard. So I spent some time working on squash, for example, and to do this, you need a genetic map. And this is the best genetic map available where you can actually put markers onto the published squash genome. The best genetic map available for squash. And if you've seen genetic maps before, the x-axis here is physical position and the y-axis is centimorgans, and these should be monotonically increasing. So you expect something sort of like this that we see in chromosome 4, these sorts of going back and forth or two segments overlapping. I'm not sure what's going on there, but it's some combination of the reference genome is messy, or there's strange recombination patterns, or the genetic map is badly estimated. In any event, it's basically impossible to use this data for IBD reconstruction. And at this point, I was sort of happy with this model because the model matches my a priori expectations based on archaeology, et cetera. And I made the mistake of actually questioning some of the parameter choices. So the second lesson here is never question your parameter choices, make arbitrary choices, and never look back, because you will only regret it. It turns out that estimating IBD chunks is actually quite challenging, especially when they're small. So this is some work from the Brownings on the power to estimate IBD chunks. And it turns out that the length of an IBD chunk that you need to be informative about a certain period of time, generations in the past, you can get from this formula. So for something about 500 generations in the past, we need to be able to identify chunks of identity by descent that are about 0.1 centimorgans. So given some sort of a rough rule of thumb of 1 centimorgan per megabase, we need to be able to identify 100 kb segments of the genome that are identical by descent. And as they show here, that trying to identify IBD segments that are really small, this is 1 centimorgan, and we're trying to identify 0.1 centimorgans. So your accuracy and your power for picking these out is not very good. And that's just because of SNP error or genotyping error or missing SNPs or or random drift giving you segments of homozygosity. So lots of different reasons. And thinking about this, I decided to look at a couple of different maize and mostly maize data sets to see, could I get the same pattern using different maize data sets, and how sensitive was the model to parameter choices. And it turns out the answer is that it's extremely sensitive to these data choices. So in this sort of analysis, you're making it requires a bunch of fairly arbitrary choices about what map you're using, how representative that map is of your individuals, how you genotype things, how you filter them, your phasing and imputation error, how you identify IBD chunks. So what size chunk do you use? How many mismatches do you allow? All these sorts of things. And I spent basically an entire summer playing around with these different parameter choices, calling SNPs from the same exact individuals with freebase versus GATK, trying different sets of maize individuals with different population structure, inbred lines versus traditional varieties, et cetera. And the short answer, as the title says, is that these analyses are extremely sensitive to data choices. So with different data filtering and SNP calling, you can recover something that looks very, very different from what I showed you, where there's no evidence of bottleneck. If you ignore the genetic map or use a bad map, you end up with no evidence of bottleneck. And this is my favorite result, that the ancestral maize population size, if you actually add up the weight of all of those maize plants, it approximates the mass of the sun about 1,000 years ago. And so that's That's probably wrong. I'm guessing there weren't 10 to the 30th different or 10 to the 30th maize plants 1,000 years ago. So you can get some pretty ridiculous models if you change some of these somewhat arbitrary parameter choices. And I started thinking this last summer, well, what if the part of the problem is short read sequencing and genotype calling with short read sequencing? And started first with maize, thinking about what if we just take whole genome assemblies, use anchor wave, which is this wonderful algorithm for aligning whole genomes to get an alignment. So this is what an anchor wave alignment of some region of the genome looks like. And from that alignment, you can actually pull out base pair differences, indels, et cetera. And so I spent part of the summer doing that. And the first thing we looked at was, well, what does this tell us? How good do the anchor wave calls look compared to the short read calls? And this was also a really disturbing result. So we know from genetic work in maize, and this is a really nice paper from Pat Schnabel's lab, where they looked at single copy sequences in B73 and MO17. So if you do Sanger sequencing, PCR, you can show that the sequence is in a single copy in both B73 and MO17, and they pick several dozen of such sequences. And then they look in the IBM RILs, and they show that you can identify RILs that have 0 copies or 2 copies of those sequences. And they map those and show that, gee whiz, what's going on is that it is indeed single copy in both B73 and MO17, but they're in different regions of the genome. And so recombination can give you these patterns. So this blue single copy sequence can recombine, and you can get 0 or 2 copies in the RILs. And then what happens with short read data, if we take one of these reads from MO17, for example, and we map it back to B73, because this blue sequence is unique in B73, the reads will map uniquely. They will look to be high quality, et cetera, and you will call SNPs in this region in B73 and say there's a SNP there in MO17. And indeed, while there may be a nucleotide difference between these blue sequences, there is not a SNP in this location in B73 and in MO17. In MO17, there's a deletion. And so we just said, with the genome assemblies, can we identify places where the short read data call a SNP, but we have a reasonably high confidence deletion in that inbred genome in the assemblies? And we do this for each of the NAM genomes. And this is work Michelle Stitzer helped with the assemblies and alignment. And some of the work on detecting error rates and overlap was done by Edwin Solares, who I think he's giving a Zevolution later this semester on some of this work. And the short version of it is that between 10 to 15 or 20% of the SNPs in any one NAM line are probably wrong. That is, we estimate 10 to 15% of the SNPs in MO17 or, in the NAM lines and MO17 in any given NAM line are probably not actually in the place where the short read SNP VCF says they are. Which is pretty disturbing if you're trying to reconstruct small IBD chunks or do anything sort of along the genome. And so I said, well, to heck with this, let's abandon the short read data and see if we can do things just with the whole genome alignments. And this has a lot of nice properties, because we can take our genetic map for maize, which we think is pretty high confidence. We can take the NAM genomes, which we know are good assemblies. We can do pairwise alignment with anchor wave, which has very few arbitrary parameter choices. We can call SNPs directly from the alignment. And then we can run MSMC2. MSMC2 is an algorithm that looks basically at rates of coalescence along the genome. And one of the nice things about MSMC2 is it also doesn't require many arbitrary parameter choices. You have to make assumptions about mutation rate, but we have mutation accumulation data for maize. So we have a pretty good estimate of mutation rate, and you have to make assumptions about generation time, but we know maize is an annual. And aside from those two parameter choices, there really aren't that many dials to turn, which means that in general, it's much less sensitive to the sort of arbitrary parameter choices or filtering things that cause problems for the IBD analysis. Still, it's still hard to get good data. I was excited by 2 recent papers this year on pangenomes for tomato and potato and said, let's do this for a bunch of different crops. So this is the MSMC2 model from tomato data. So this is using just Cerasiforme and a number of Cerasiforme genomes aligned. Again, on the x-axis here is generations before present. The dotted line here is around European contact, and the y-axis effective population size. And you can see that the model, each line is a different chromosome for tomato, and you can see that there's tremendous variation among the chromosomes, and there's not a lot of resolution in the recent past. And even in the distant past, there seems to be variation among chromosomes. And when we looked into this and thought more about it, it almost certainly because there's quite a bit of foreign introgression into tomato genomes from other species, Pimpinella folium, et cetera. And this also leads to, if you start looking at things like heterozygosity along the genome, you see segments of really ridiculously high heterozygosity because some segments are carrying entire chunks of a chromosome from a different species. And there are very few of these that are fixed. They're all over the place among these genomes and from different wild relatives. And in theory, you could go back and identify each of these chunks individually in each of these genomes and mask them. But that I made some stabs at doing that. And it turns out to be difficult and decided it wasn't really worth my time. And so we said, well, let's look at potato data because there's lots of good genomes. There's this nice pangenome for diploid potatoes. And if we run the model for potatoes, we see that basically we have 0 resolution in the last, say, 10,000 years. And this is because for potatoes, we actually have no idea what the generation time is. And we had, Michelle and I had a very nice conversation with Steph DeHaan at SIP, the CGIAR Center for Potatoes, asking about potato varieties and sexual reproduction in potatoes and what generation time is. And the short answer is nobody has any idea. That most of the varieties are, of course, clonally propagated. Some of the varieties, or many of the varieties, seem to have been around since European contact. So there are varieties that were created by the Inca and have lasted or have hung around for 600 years. So maybe generation time for those varieties is 600. But there are a few varieties that have been created since then. On a sort of interesting note, in talking to Steph, although there isn't good published data on this, his hypothesis is so there's something like 4,000 different traditional potato varieties. And his estimate is the vast majority, 90% or more of those, have been around since European contact. And so his hypothesis is that actually Inca and other groups may have had active breeding programs to create new potato varieties, and that European contact basically led to the demise of these breeding programs, such that there have been almost no new potato varieties developed since then. And most of the traditional potato varieties that exist today are ones that were created during by the Inca Empire. And there certainly are archaeological sites like this great, this site here, I'm blanking on the name, I think it's Moya, but where there's clearly a breeding center for Inca where they were testing different varieties from different locations. So there is clearly some active breeding activity. And although there isn't apparently great data for that hypothesis, I really like that idea. But the short version is it's still hard to get good data and get meaningful results. But we have good data for maize. So we took the NAM parents, we aligned them, we can run MSMC2. And what we get out is a really beautiful result that is entirely consistent with our initial IBD model and the archaeological data. So here, each line is one of the 10 chromosomes of maize, because this takes a ton of RAM to estimate. And we see a decline in population size associated more or less with the timing of maize domestication about 9,000 years ago, and another dramatic decline in population size associated with European contact about 500 years ago. So this was really exciting. And adding to this, we decided, well, let's see if an entirely independent model can give similar results. So we took site frequency spectrum data. So we just get counts of how many SNPs we see in these whole genome assemblies and their frequency. And we take that and working with Silas Titus, We put those into a program called Mushy, which reconstructs the demographic history from the site frequency spectrum. And we get qualitatively a similar pattern. So the timing of the domestication bottleneck seems to be a bit off, or at least different. But we see a really nice association between a decline in population size with European contact. The relative sizes of these declines differ between the two models, but qualitatively both say there was a dramatic bottleneck associated with European contact. And so this was really exciting. I think qualitatively, although I believe this result, well, I'll end with the caveats in a sec. I think this is an exciting result, and qualitatively, I think that this is right, especially given all of the archaeological literature. And if so, I think it does have implications for thinking about crop diversity. So this is a fairly famous picture that I forgot to put the citation for, from Steve Tanksley, who who first proposed sort of this idea of how crop domestication and modern breeding could lead to a loss of genetic diversity, and showed this model where you have a number of, I think it's 4 or 5 different alleles in wild species. A few of those are lost by selection or drift in early domesticates, and then more are lost in modern varieties. So this idea of consecutive bottlenecks. But this cartoon, although it's been widely sort of represented or copied, doesn't really do justice to what this model is suggesting. So if you simulate data under this model, you get something more like, for a 10 kb region of the genome, you get something more like 70 different alleles. You get a wide variety of alleles in any reasonable size sample. And the vast majority of those, so we're not losing 1 or 2 alleles or half of alleles, we're losing more than 70% of all alleles in traditional varieties due to a dramatic bottleneck associated with European contact. So if this model is right, it potentially has really dramatic implications for allelic diversity present in traditional varieties and crop wild relatives compared to, in crop wild relatives compared to modern maize, because of this loss of diversity associated with subsequent bottlenecks. But the last lesson that's worth thinking about, in addition to not paying attention to other disciplines or questioning your parameter choices, is to never ever take a hard look at your model and ask whether or not it's any good. I always suggest when there's when people have estimated a demographic model, you put your data into software, you will get a demographic model out the other side, and that doesn't actually mean that the model is really any good. And I think it's really a requirement to simulate data under that model and ask how well does that simulated data recapitulate your observed data. And it turns out that for the model that I've estimated for MSMC2 or Mushy, the answer is not so great still. So the black line here is a density plot of genetic diversity, or pi, simulated under the model, and the red line is the observed mean across chromosome 7 in maize. And we do okay for the observed data. It's certainly within the distribution for simulated, although if you plot the densities, the peaks aren't the same. But if we look at Tajima's D, which is a measure of a site frequency spectrum, we get something really dramatically different, where data simulated under the model suggest a dramatic loss of low frequency alleles or increase in intermediate frequency alleles, and the observed data actually are consistent with a weaker loss of low frequency alleles. And so there's this disconnect between the site frequency spectrum of the observed data in the NAM genomes and the simulated data is somewhat concerning. And although, as I said, I qualitatively believe that the model is right, I now have 3 different models that are telling me there's evidence of a recent bottleneck. The exact strength of that bottleneck and the details of the model clearly aren't right, because they aren't able to recapitulate the real data. My best guess right now, and I don't have an answer for what's going on, my best guess right now is that the simulated models or the estimation procedures are assuming that your data is neutral. And if you looked closely at one of my previous slides, what we've done when we align all of the NAM genomes, the models require that you have no missing data. And so rather than impute data that we know is not there because there's a deletion in one of the genomes, we only take base pairs where each one of the NAM genomes has a base pair at the same position as B73. So we require all 26 NAM genomes to have a base pair, a alignable base pair at that region of the genome. And on average across chromosomes, this is something like 5% of the chromosome. So only 5% of the base pairs on any given chromosome actually have an alignable base pair. That's not to say the same nucleotide, just a base pair that can be aligned between across all 26 NAM. And that number, of course, will increase as your sample size increases. Now, this isn't maybe quite as woefully terrifying as it at first it seems, because most of those base pairs are alignable in the vast majority of NAM, and just one or two of the NAM parents have an indel or an inversion or some other structural variant that means that it's not alignable across all 26. So most of the deviations from this are rare, but across all 26, it gets down to only about 5% of the chromosome. And it's probably pretty unlikely, my guess is that 5% of the chromosome is evolving neutrally. That is probably a conserved part of the chromosome. And so our next, or the efforts that I'm trying now are to try to think about estimating models that allow for background selection or conservation of sequence and demography at the same time, although that's quite a bit more difficult to do. So that's where we are. I think qualitatively that the models are right. Their predictions aren't very good. The short read data is somewhat disturbingly bad, and aligning from whole genomes is consistent or argues that there's really a ton of structural insertion deletion variation and lots of and very few sequences that are conserved across all domains. So I'll end there on that, maybe not the highest of notes. Lots of folks to thank, both in my lab. Michelle has done quite a bit of work on this and helped me with the whole genome alignments. My go-to archaeologist, Dolores Paperno, who gave me some of that archaeological data and pointed me to a vast literature of archaeologists saying, yes, this is obvious. Other folks in Cornell who helped with anchor wave, Aaron who helped with mush, and Silas who helped with the mushy analysis. And I'll end there and happy to take any questions. Let's see, I think there's some in chat already. Yes, so Ed's saying that's why IBD chunks, or the variation in IBD, is this what you're saying, Ed, that the sensitivity of IBD is why 23andMe results jump around so much?
Yeah, I mean, and I remember Svante Pavo, who was mentioning it to me, he said, yeah, don't believe the Neanderthal things at all. Every time we change the parameter a tiny bit, it'll give you a new Neanderthal proportion.
Yeah, and I mean, this is the problem with estimating ancestry from so long ago and introgression from such a long time ago, is that you're really trying to identify small chunks, and so it's very sensitive to any of those parameter choices.
And you know, even though if you look at our generation time issue between corn and humans and Neanderthal and corn, and where you're kind of going, we're talking almost probably same ballpark number of generations or something like that.
It's actually European. So European contact and Neanderthals is very different. Neanderthal contact is about 5,000 generations ago.
Okay.
I think it's about 5,000. So it's pretty similar to the timing that we're thinking about for the Mexicana introgression stuff.
Okay, cool.
Maud asks about LD approaches. I have been very skeptical of LD approaches. There are a number of LD approaches that can, there are a number of approaches to estimate LD in the recent past, but these folks, Garcia Ortiz, that looked at this indigenous American populations, actually estimated with IBD and with LD approaches, and LD approaches showed absolutely flat NE in the recent past in humans. They weren't using exactly this approach, but I have seen, yeah, I haven't
similar,
tried
you think? Okay.
So, yeah, but I think
Mhm.
and it's a little bit, it should be a little bit more robust than IBD because you're not trying to identify start and end of blocks. But it still seems to be sensitive to things like data filtering and Hardy-Weinberg, et cetera, and error, genotyping errors, et cetera.
Okay, okay. Okay. I
Um,
had another question, but later, but I don't know if I can ask it now.
yeah, go
or-
for it. Go for it.
Yeah, it's just that how do you reconcile then that to the model that you propose? Like you showed a slide at the beginning from the Bessinger paper, I think. And do you think that this second bottleneck in a way is confounded with the first domestication bottleneck, and so that the domestication bottleneck in the model you propose is like overestimated? Or because you also found that the data were consistent with a huge exponential growth, and so if you had a very, very strong bottleneck that was recent, then maybe you shouldn't have or this excess of very rare variants. So I am not exactly sure
Yeah, right.
how.
So the question is, how does this confound sort of previous estimates of bottlenecks?
Yeah.
I think that's a great question, and I think it would depend on the data type and the method. So trying to do anything from the SFS with a small number of samples, which is what we did in the Beisinger, you probably have no power to say anything in the relatively recent past unless there was something super dramatic, and maybe there was. So I think it depends on your sample size and the approach, but it certainly could. Yeah, it's certainly possible that it could, and I think that that's a fair concern. At least in our MSMC2 and Mushy models, in both of those, we do identify two separate bottlenecks, and in neither case, you're not telling it there's a bottleneck in either of those places. So both models seem to suggest that there were two different bottlenecks, but the relative severity of those and the relative extent of recent exponential growth, I think you're right, is potentially confounded, and we don't have a good model yet. Let's see, Matt, we looked at teosinte. Yeah, Matt, so you may remember that we argued about this a year or two ago about what our predictions would be for teosinte and whether we would expect teosinte to show changes in population size that mirrored maize or that would be opposite of maize. So you're arguing that fallow fields could be fertile ground for teosinte colonization, increasing any. I think an alternative expectation would be that because teosinte is a ruderal and needs disturbed habitat, that as those fallow fields regrow into forest, teosinte population size could actually decline. We did look at using the IBD approach. We did look at a number of different teosinte data sets. And in general, the teosinte, at least with the IBD approach, the teosinte data largely mirrored the maize data over this time period. There did seem to be a bottleneck associated with a bottleneck in the recent past in the teosinte as well. I'm not super confident on that. The teosinte data isn't as good as the maize data, and the IBD approach, as I said, is woefully sensitive to stuff. But that super preliminary result suggested that actually the teosinte data may parallel the maize data. One thing to keep in mind, and one potential reason why the model doesn't predict the data well, is I'm modeling a single population with no gene flow, no population structure, and just demographic change over time. So things like hybridization with Mexicana 5,000 years ago, or more recent gene flow from teosinte, all these sorts of things are not being modeled and certainly could have an impact on diversity data in the present. Let's see, Jan says, is it possible to use currently available data sets to estimate how much of the loss of alleles during domestication breeding are beneficial to agriculture? That's a good question, and I think it's hard to do with PopGen data alone. So, for example, we can use models of phylogenetic conservation or population genetic models to identify alleles that are putatively deleterious. That is, that have been conserved over evolutionary time, or windows that show evidence of purifying selection in the present. But it's hard, I don't think from that data alone, you can make claims about an individual allele and whether it's likely to be beneficial to modern agriculture. So we can show, and we've shown in the past that in general, as a rule, alleles that are conserved over phylogenetic timescales are mostly deleterious and do mostly decrease yield. I think that's a relatively safe qualitative claim in general. For any given allele, I wouldn't put any money on how well the PopGen data or are able to predict that. So, for example, if we compare phylogenetic models to more recent PopGen models, we see we see a few cases anyway, where something that appears to be strongly conserved over phylogenetic time is no longer conserved in the recent past, which could be consistent with a shift in selection. And none of those models, I think, are really able to reconstruct something as recent as the last 100 years of breeding. And so I wouldn't put any faith in any of those and would say you need to test those alleles in actual plants in the field. Ed, Ed, you agree with about 5%? 115 per core gene. Yeah, so I think it's a little bit more than just the exonic base pairs of each gene. I think it's about twice the size of the exonic base pairs of each gene. But it's basically genes and important regulatory stuff is my guess.
Yeah. No, I mean, the average CDS is 1,000 base pairs. So yeah, I think that's about the right number, I think, to align.
Yeah, but is disturbing nonetheless.
Yeah.
Let's see. And then more discussion about the Beisinger model. Yeah, and the Beisinger model also didn't do a good job of it was very noisy with maize data unless we constrained teosinte to have a constant population size, which is also a concern. It is worth noting that two different MSMC models based on short read data in papers that we published with Matt are also consistent with at least a gradual decline in the last 1,000 years of population size. And whether or not that's the European bottleneck or something else, I'm not sure. So I think qualitatively, the genetic data suggests that there probably was this extra bottleneck that's largely consistent with tons and tons of archaeological data. But I think I'm not quite at the place where I'm willing to bet any money on the specifics of a given model until we can recapitulate the empirical data. Let's see, Wojtek, you had your hand up.
Yeah, this is a technical question, and I'm not sure if you've looked at that. So I was thinking about this awfulness of the short read data, and in some cases we would have genetic data to implement an LD filter, and I wonder if you tried it and if that helped.
We didn't here, but in the HapMap3 paper from Ed's lab, an LD filter was actually one of the best ways to filter out erroneous SNPs. What we did look at there is it turns out a large number of those SNPs are in low copy TEs. So they're in TEs that are either old enough that they've diverged among the NAM genomes or are single copy or low copy. Such that reads from, say, Mo17 will map uniquely to B73. If you mask if you take the TE annotation of maize and you mask any SNP that overlaps a TE, the vast majority of those erroneous SNPs go away. So I can't remember, 80 or 90% of those are inside of a TE, suggesting that if you're willing to just ignore all of the TE space and that 80% of the genome you just ignore, you can solve a lot of this problem. Fausto.
Hi, Jeff. It's very nice work. But I was wondering, your demographic models should depend on the sampling. And there in the Browning data, you can see that the decline in population size in the American populations in Mexico is not that notable as in the other places. So I think it's given because there was more mixture between there was not how can I say this? In the Americas, there was more extermination of the natives. In North America, there was more extermination of the natives population than in Mexico or in Peru. So it seems that that it is if you sample like North American maize, there should be different demographics than Mexican maize.
Yeah, you're entirely right. If you look at the human data, the decline in any indigenous populations, for example, in Puerto Rico, it's about 95%. And in Mexico, it's closer to 50%. So there's dramatic regional variation in the decline in indigenous populations. And so we might expect the same to be true for maize. And this is actually one of the reasons why I first started playing with the IBD model and looking at different data sets. So I took data from a single farmer's field in Guerrero, or I took the 282, or I tried with different data sets, expecting that I would hoping to see different results for different regions, and instead discovering that I get total craziness that depends on how they called SNPs and how they filtered the data. I think Michelle and I have gone back and forth on this. I don't think we actually have good enough data yet from a number of South American genomes, for example, genome assemblies, or enough individuals with WGS data to do this really well. But it's certainly something that we're thinking about, and you're entirely right, would be good to do, and would be nice to show that this varies across regions in a way that's consistent with variation in indigenous human populations. Marcus?
Yes. Well, thanks for this really interesting talk. Have you considered sort of the opposite assumption, right? Because you have the strong relaxation of selection of keeping maize or whatever crop, and having the potential for a lot of gene flow between the wild ancestor or here teosinte and maize. Maybe less in maize, so right, maybe the extinction is there true because it really does not grow well without humans. But in other crops, you might have just a strong relaxation of selection and potential for very strong gene flow from the wild relatives, which would sort of do the opposite. But so following up what you just said, you should see a difference then in South America and Mexico, right? Where in Mexico you would have that potential for gene flow from teosinte, but not in South America.
But you're suggesting that European contact then changed patterns of gene flow with wild relatives?
Increased it, right? Because there's less people taking care of that maize, a little bit falls on the ground and intercrosses again with teosinte and then.
Maybe. So this, for example, this is something that Steph brought up with potatoes, that if you leave a potato field fallow, you can come back 5 years later or 10 years later and still recover some of those plants. That's certainly not the case with maize. I think it would, as you say, it probably would depend on the crop, which is part of the reason why we were initially interested in trying to do this with multiple Mesoamerican or American domesticates, to say, do we see the same pattern, and does the pattern match what we know about the natural history of these species? And ended up being so mired in it's hard to do with the genetic data that at least for the near future, I'm not interested in following through with either the potato or tomato data, and I'm not aware of whole genome assemblies for bean, for example, that will let us do this.
Right, but even if you don't see the same, right, it might be just because they can actually interbreed with something more feral.
Yeah, no, right. That's my point.
Maybe that's less the case, but it might still be the case that you get more introgression from teosinte again because of less selection for those
My hypothesis is that's not the case, but that difference in natural history among species is why, ideally you'd like to look at, beans and tomato and maize from the same region, from a couple different regions, and say, do we see the regional difference and do we see a difference between species? I think that would be the ideal setup, and I'm not aware that we have data to do that.
Yeah, between species, between regions probably would be very interesting, right? Because you expected this effect, you would expect to be very different outside of the distribution range of the ancestor or of the wild relatives.
Right. Or as Fausto pointed out, the differential decline in the population of farmers.
Yeah, right. Thank you.
Fausto, did you have another question or? Okay. I think I caught everything in the chat. Yeah, well, thank you, folks. If folks have suggestions about other things that alpacas and llamas show higher gene flow, hard management was abandoned. Interesting. If you have suggestions about other things I can do or other things to play with or other data sets might be available, we've talked to folks about sunflower data because there are a number of genomes and they're currently playing with anchor wave and sunflowers. But if you're aware of other data sets or other approaches, that would be really fun. Please let me know. Thanks all. And I think, Carl, we have somebody lined up for next week, right?
Yep. Nicole Choquette from the Holland Lab for next week. So thanks everybody for coming.
Right. Thanks, folks.